Thank you so much, Dr. Melendez, for the kind introduction and more importantly for the invitation here to San Juan, Puerto Rico. Just like Eliana, it is my very first time here in Puerto Rico, and all I can say is I'm just loving it. And before I really brace for some snow in Nebraska on Sunday, so kind of really enjoying the weather though. So like she mentioned, I have been doing a lot of work as part of my postdoctoral work in neuro-AIDS. And then I've also started to work at the interface of neuro-AIDS, HIV interaction with drugs of abuse, specifically methamphetamine abuse, and how these two factors can sort of lead to accelerated neurodegeneration, specifically in HIV-associated neurocognitive disorder, or HAND as we call it. I just changed the title a little bit because when Loida Invite kind of contacted me, I was sort of trying to talk more about the systems biology, which I think we'll talk, but given the fact that this is a proteomics sort of some mini-symposia, I will try to stay specifically focusing on the proteomics aspect as such, and accordingly I've tried to label my entitle my talk as an omics perspective to decipher the synapse. This is the current research which I'm actually doing, and as she mentioned, my other arm of my research very recently which I got into is also into autism, specifically looking at maternal immune activation. So whoever is more interested is can talk to me later, and I'm not going to be talking anything about the autism research today. Like I said, this talk, most of the studies has been published, and most of the studies that you will be seeing have utilized omics approaches, specifically proteomics and metabolomics, and these have been from a discovery standpoint as such. So, which is very critical before one really needs to, gets further going into understanding the biology associated with these identified hits. And what do they actually mean in the context of a disease, which is very important. So just to give you a little brief introduction, I'm sure the audience here are sort of a mixed bag if I get it, but you probably have a very strong HIV research program going on here at UPR. So we are majorly interested in looking at the infection of HIV in the brain in a more chronic setting as such, and how it actually leads to neurodegeneration. So this is a very simple schema which I borrowed from one of Avinath's reviews, where the infected, where the macrophages basically which are infected with the virus, they enter into the brain, they pass the blood-brain barrier in the process, breaching that, and basically activate a cascade specifically by the release of chemo and cytokines. So this particular release is mediated through the activation of glial cells, specifically microglia and the astrocytes, and then since these are activated, these chemocytokines basically then go and destroy the neurons. So just to tell you that the HIV does not infect the neurons, but the death that we see in HIV is due to a secondary cascade mediated by these glial cells and the release of these chemo and cytokines as such. So where do we stand in HAND is the question that a lot of people are currently looking at. So HAND, or HAND, is also referred to as HIV-associated neurocognitive disorders. It encompasses a group of cognitive and behavioral changes and comprises of three major classes. One is asymptomatic neurocognitive impairment. Then you have the minor cognitive motor disorder, and then the severe form of neuro-AIDS, which is known as HIV-associated dementia. And now since most of the HIV-infected individuals are on antiretrovirals, the further enhancement in the field of combined antiretroviral therapy has actually changed the demographics, specifically in HAND. So while we see the most severe form has actually declined, people are actually still getting demented, but then you have an increase in the milder, less profound cognitive impairments, which are actually on the rise. So though the longevity of the person infected with the virus and who is on antiretrovirals, he's living longer, the quality is actually not really changed. He's actually not getting better as such. So this is a little snapshot of where we actually have. This is, like I said, the HAND and the pyramid here is, and you see on the very top is a very small fraction of people with HAD. It's almost like 5 to 7% these days, which is very low compared to about 25 years ago, which was about like 35-40%. And this is just the data showing, the bar, how it's actually changed before the advent of the retrovirals versus the the use of retrovirals these days as such. So pathological correlate of hand or neuroAIDS is basically referred to as HIV encephalitis in humans. So it is very easily recognized, when one does the immunostaining with HNE, you see a characteristic pattern called as the MNGCs, or mononucleate giant cells, basically of the monocytic lineage. And you also have a nice staining for P24, including the activation of microglia as assessed by immunohistochemistry using this particular marker CD163 or IBA1. So when I started off working in this field of neuroAIDS, proteomics was on the rise at that time, about like 6-7 years ago. And basically, we wanted to utilize an omics strategy where we can kind of get a snapshot of what exactly are the changes occurring in the brain as such, and with special emphasis on HIV-associated neurodegeneration. And then, so there are different levels that one could actually sort of understand or try to gain insights into this. People, there is genomics, transcriptomics, proteomics, but then, we see that a common goal of all these approaches is basically to identify potential pathogenic biomarkers as such. So when I say biomarkers, I still get very nervous these days because it's a very broad term and people kind of get a little iffy. It's like, is it really a biomarker as such? Considering the fact that people who work in clinics or, with human subjects, the biological variance is so high, and then from cohort to cohort, there is a huge difference as such. So that is why in my talk, I will kind of mellow it down and keep it to more as potential pathogenic markers rather than saying biomarkers as such. And my major emphasis is going to be on the application of mass spectrometry based proteomics and metabolomics, which actually have been very promising in the field of neurological disorders as such. So like I said, I'll be talking on mass spec based omics approaches, showing you some data which have all been published, where we have used biofluids, specifically the cerebrospinal fluid, the plasma, and then the current work which we just published in the brain, which we call as organelle proteomics as such. But the bottom line is, whatever the approach one uses looking at either of those compartments, whether it is the biofluids and or the brain, the idea is basically to identify it as potential pathogenic molecular markers, and more importantly, learn about the mechanisms associated of how these identified hits can help us understand the ongoing changes during the course of neurodegeneration as such. So this is for those who are interested, we have sort of summarized our works in the field of neuroAIDS in one of these review articles in Genome Medicine published about a couple of years ago. And I'm going to be starting off showing you some published works in using the cerebrospinal fluid as such. But in the interest of time, I would not be talking about the metabolomic approaches, which we have actually studied. This was actually published about 4 years ago, and this was actually one of the very first studies using a global untargeted mass spectrometry based proteomics to identify potential metabolic markers during the course of SIVE induced CNS disease as such. So our model systems are very critical to understand the ongoing pathology. So my postdoc mentor, he's an expert in the field of using the rhesus macaques and specifically infecting them with the SIV or simian immunodeficiency virus. So the SIV is the closest relative known to HIV, and it induces AIDS similar to HIV-1 in humans. it produces clinical syndromes similar to that of humans, and it is one of the most premier models of HIV neuropathogenesis. But again, the downside is that, it's a very monkey business is expensive, so you need a lot of funding to kind of work with such heavy duty model, which I call it. And so our major emphasis was to identify potential molecular markers which could actually help us understand gaining vital insights into the ongoing pathogenic process during the course of neurodegeneration as such. So this is one of the papers which we published utilizing CSF. So the problem with the CSF is that, or let me start the other way around. The reason we chose to look at CSF is because CSF is a pretty nice window. Given the fact that it surrounds the brain and the spinal cord, it acts as a nice window to understand the ongoing processes during the course of neurodegeneration. And specifically, when one wants to look at a longitudinal sort of a study where you want to take different time points during the course of disease progressions, CSF does play a very important role, and it can be easily obtained. Easily obtained? Well, yes, but not the amount of quantity that one can actually get. So we asked a very simple question like, is there a way that avoiding extensive fractionation, can we still utilize minimal amounts of CSF and still identify potential pathogenic molecules? So we started off using a bare minimal amount of CSF. Again, this is monkey model, so we couldn't really get much CSF, and whatever the CSF that we got was a very, very clean tap. So we chose 100 microliters of the CSF, and then basically did a very simple approach where we used a one-dimensional approach, and it was an unlabeled or non-labeled method of quantitation as such. So we identified potential hits, and we further validated these hits since we were dealing with very small amounts, and with a one-dimensional approach we wanted to really be sure what we identified is indeed a true hit or not. So we did analysis and we identified them using RT-PCR as well as immunohistochemical staining. And just the take-home message from this particular study is that we still can actually use this particular technique where minimal amounts of biofluids are actually available. So just to give you a little idea of what the proteins we identified. So these were the same animals before and after infection with the SIV, and all these 4 animals actually had encephalitis, which I said is the pathological correlate of neuro-AIDS as such. And we identified a bunch of proteins which were actually upregulated, and the minimum criteria that we had was at least 2 unique peptides being identified, and we saw a bunch of these proteins, and we also set a cutoff basically at about 2-fold higher or lower, and this is what is actually represented here. And we further validated the expression of these identified proteins looking at their RNA expression. So we chose the hippocampus, and because we didn't really have the caudate nucleus or the caudate plexus as such for this sort of analysis, so we validated them. We could only validate the upregulated proteins, and there was no it was pretty difficult to look at the downregulation. So and this is the representation of these 5 upregulated proteins that we actually identified. This is represented here in the log scale as such. And we also chose an independent set of animals where we validated these 5 different proteins, and we see that there is, in all these animals, we see a nice validation of what we actually see from our initial screening using mass spectrometry based analysis as such. So this is one example of one protein, the plasminogen, which we also looked at using immunohistochemistry. A and B are the control monkeys without any infection. C, D, E are basically the monkeys with encephalitis. And then we also looked at the expression of this particular protein in the choroid plexus, where the CSF is actually produced. And we see the upregulation of this particular protein here in the encephalitis monkeys versus the controls as such. So these, and here I would like to highlight a couple of papers from Loida's group as such, where they have actually used human subjects as well. And I think this is one of the papers published before us, where they looked at the CSF-Proteomic fingerprints from people with HIV infection and people with HIV-associated dementia. And I think one of the proteins you guys identified was the copper zinc SOD, one of the antioxidants. And then Powell's group, where Loida is also one of the authors on this, they identified certain other proteins from the cerebrospinal fluid, but here they used quite a bit of amount of the protein. Here they pooled the samples. In our case, it wasn't actually the case. We had individual monkeys, individual CSF, and basically analysis of those individual samples as such. So then we actually asked that, CSF is great, but then, the mainstay of diagnostics is the blood, the plasma as such, because it's relatively much more easier to obtain, even in terms of the quantity as such, compared to the CSF. And but the downside again is that depletion is a prerequisite, given the fact that albumin and immunoglobulins basically take up almost like 85% of the total protein content, and hence if one needs to look for these potential markers, we need to basically do a depletion in this case. So in this, we applied a double immunodepletion, where the first column depleted the 14 most high abundant proteins, and then in the second one, the column which was in tandem to it depleted 77 medium abundant proteins. And basically the flow through that we actually have, which are the lower abundant proteins, were the ones which we used for eye-track analysis as such. So employing the similar strategy, again these were the same 4 animals where we did the CSF. So this was again the same plasmas from these guys, and I think we had 2 more extra animals at this time. And we found one of these proteins is afamine, which is a vitamin E binding protein to be downregulated in SIV encephalitis. So here is a quantitation. As you see, these are animals with encephalitis, and these are just SIV infected animals with no encephalitis. The reason I'm showing you this is that people wanted to see that whether the virus itself is causing any change here in the brain, or is it due to the manifestation of the disease that actually results in this particular difference as such. So as you see that just the viral infection does not cause, there's no difference in this particular protein, but it is very specific to CNS associated degeneration during the course of disease progression, as seen in here in the encephalitis animals. So this study was very nicely supported by another study from Powell's group, where they actually used human samples. Basically, again, these were HIV-associated dementia patients versus just HIV positive. And as you see, here is afamine. Here it is basically downregulated, similar to the ones that you see here in the monkeys as such. So this is one of, so here I could kind of say with a little confidence that, yes, this is a potential biomarker for neuro AIDS as such. So moving forward, the next thing that we wanted to kind of look at was, how does the virus, in terms of in synergy with methamphetamine in drugs of abuse, and specifically here is meth, cause to HIV-associated neurodegeneration. So just to give you a little snapshot, drug abuse is a very significant comorbid factor in neuro AIDS. And our research basically focuses on meth abuse, and meth abuse has been increasing at a very alarming rate. It is one of the most highly abused drugs in Nebraska as well, and the fact is that it is very cheap, and it can be pretty easily made. Now don't ask me how to make that one. There are several secret labs that actually do prepare meth, and so anyways, so the point is both, compared to the HIV positive only versus METH positive only, people who abuse, people who are HIV positive and are METH abusers, they are actually more prone to, neurological dysfunctions, as such. And coming to the studies that have been done in this particular areas, there have been very limited as such, and hence understanding the synergy of how these two lead to CNS dysfunction is very critical. So in this line, we again used our SIV monkey model as such, and we started off looking at the plasma markers. So this was one of the papers which we published about a year and a half ago, in 2010, 2 years ago, and we found one of the proteins is EC SOD or SOD3, which is an antioxidant, and we also did a targeted approach, and we found that one of the proteins, glutathione transferase, which is also an antioxidant as such, to be upregulated in the monkeys treated with infected with SIV and also treated with METH. And here I would just like to point out a little caveat or limitation that we have. Like I mentioned, monkey business is expensive, so we did not have two other groups, basically control monkeys without any SIV or drug and only METH only group as such. So that is actually missing in this, but still the point was we wanted to see whether the drug in conjunction with the virus does have an impact or not. And this where all these monkeys were chronically infected for 23 weeks with SIV and then 19 weeks of methamphetamine treatment as such. So the total regimen lasted for 42 weeks. And we basically collected the plasma before the animals were actually sacrificed as such. So, what is this? Two duplicated. Okay, sorry. So now coming to the main emphasis, where I really want to look at the brain. While biofluids are really excellent source to identify potential markers, the real changes are actually in the brain. So to understand that, we started to look at the brain per se as such. So here is this is one of the very recent papers that we actually published in PLOS ONE, and we found one of the proteins is this PDIA3. It is a disulfide isomerase which is upregulated in these in the METH-treated SIV and METH-treated animals versus the SIV-only animals as such. And since this is a genome, it was a gene array, and I wanted to very specifically put my talk focused on proteomics as such, I wouldn't be talking about this thing today, but I'll be happy to discuss with you, whoever is interested. So, and again here I should say there was no extensive fractionation or anything done. It was just the total brain that we took, the caudate nucleus specifically, we mushed it up, we isolated the RNA, and then basically performed a straightforward microarray as such. So as a biochemist, I then thought, we really need to understand what is really, what is actually ongoing at the level of the synapse. Why the synapse? Because this is the business end of neurons, and as you see that, basically these are the synapse is actually this area where two neurons communicate, and in fact communication is left. Just imagine we have nobody to talk to, the amount of impact that it actually does on ourselves is pretty high. So that's why it is very important that in the brain too, that the neurons sort of communicate to each other, and that's the junction where called as the synapse, where these sort of two neurons sort of exchange information. And I just want to say that neurons that actually wire together, they actually fire together. So this implies so how can actually one study the synapse? Is there a way that one could actually look at it? Yes, as a biochemist, I basically wanted to utilize my graduate expertise in isolating synaptosomes, which are actually pinched off nerve endings, and they actually contain all the pre and the post-synaptic components. And the important aspect is that these synaptosomes, these are enriched in receptors, transporters, and ion channels, and these are very critical regulators of neurotransmission, which is the source of communication between two neurons in the brain, and are very important for synaptic plasticity, which regulates a lot of processes, especially behavior, emotion, cognition, etc. as such. So big question that one has is that, whether to use a fresh tissue or one has to use an archived tissue. So the other thing is also the purity of the synapse that one actually kind of can obtain. So it is very easy to get using any of the rodent models, getting fresh tissue is pretty easy. But given the fact that we are dealing with a disease scenario, specifically like HIV, which is a very chronic disease, and also drug abuse as such, getting the brain fractions from and people who are interested in in translational medicine, it is very difficult to kind of get brain tissues as such, unlike the biofluids. So we were thinking whether one can actually utilize post-mortem tissues or ones which are archived tissues and still be able to use this as a model to understand the ongoing changes of the synapse as such. So this is I borrowed this slide from Rudy Ebersold's paper in Proteomics, but published about 7 years ago, where they actually do, this is an EM picture of a purified synaptosomal fraction. You see that this is a presynaptic neuron filled with synaptic vesicles. You have intrasynaptosomal mitochondria, and this is the synaptic junction. So we have actually a wonderful brain bank at our METH center, and then just out of curiosity, I just picked one human brain and then basically performed the similar analysis. I isolated the synaptosomes, performed EM analysis, and as you can see here in this in the little inset here, we could basically see a nice presynaptic terminal loaded with the synaptic vesicles and also the intrasynaptosomal mitochondria as such. We further analyzed the fraction basically to look at how pure the preparation was. So we specifically used one protein called as SNAP25, or synaptosomal associated protein of 25 kilodalton. So as you see, compared to the homogenate, you see a significant enrichment of this particular protein in the synaptic fraction, suggesting that this isolated synaptosomes are pretty vital sources to basically which one can actually use to understand the ongoing changes in the brain as such. So here is a little schema, and I think this is what I was actually discussing with Loida's group as such yesterday. So this is a very straightforward, it's a very simple straightforward process of isolating synaptosomes. You might, because I've been doing it for the past 12 years, I think it's pretty easy. But for those who are just getting into this thing, it is challenging. It is challenging. I don't want to dishearten you guys, but it is that way. So basically, you can take the brain region of your choice, what you want to look at. And in our monkey study, we actually chose the caudate nucleus, which is part of the striatum. And why the striatum? Because it is a target of both HIV, SIV infection as well as methamphetamine as such. So this is, you take the brain region of interest, you homogenize it, and then basically you do a low spin to collect the post-nuclear supernatant. So this post-nuclear supernatant is further processed at a low speed as such to get the crude synaptosomal pellet. So this particular pellet this can biochemistry can be tedious at times. So this, in case one wants to stop, you still can store this synaptosomal pellet at 4 degrees for overnight, not beyond that, please. Especially for people who are interested in doing uptake assays, people do use this sort of a methodology as such. And then since we wanted to make a more purer fraction, we further went ahead and performed a sucrose density gradient, where we did high-speed ultracentrifugation, and then we basically got the synaptosomes as such. So how do they look like? So this is how they actually look like. The main challenge here is casting the gradients. This is a very cumbersome process, and you have to make sure that the gradients do not break, especially when you have, if there is a lag time, like for example you want to move from one point to the other, I would highly recommend people not to really carry them after casting, but go to the place where you think the ultra is available, cast your gradients over there and try to keep it at 4 degrees till you're ready to get loaded, you're ready to load your samples on the top. So this is how it actually looks like on a sucrose density gradient. So you basically get the synaptosomes at the interface of 0.8 and 1.2 molar sucrose. So we give it a wash briefly, And then the purified synaptosomes are dissolved in the physiological buffer, which is 0.32 molar sucrose, and one can from there, after precipitation, to get rid of any salt or any other contaminants, we basically do a precipitation with acetone as such, and that precipitate, you protein estimate, trypsin digest, and then you can actually do your downstream MSMS analysis as such. So utilizing this schema, we went ahead and we did our monkey samples, and this was a paper which we just published in May, and we identified a total of about 1200 proteins, of which we found about like 800 proteins which were differentially expressed. And then one cannot look at all these different proteins, so we had to use a certain cutoff as such. And then one protein that we basically focused was this ATP1A3. So this is a neuron-specific protein, and it belongs to the sodium potassium ATPase family. It is a complex, this particular family is a complex family of integral membrane proteins that is very important for maintaining the ionic gradients as such. And there are 4 alpha forms and the 3 beta forms, and then the one that we actually found is the alpha 3 subunit, which is exclusively neuronal, and there is emerging role as a signal transducing molecule for this particular protein as such. So we wanted to validate it in vitro. Of course, we cannot have cultures from monkeys or anything, so we chose a rat model basically to look at whether the initial observation that we see is true or not. And you see here is that this is the control cells, these are the METH treated cells, and then there is no difference in MAP2, which is a specific neuronal marker, but you see when you converge them, the amount of expression that you actually see is of ATP1A3 is much higher in the METH treated versus the control. So we also did use a specific presynaptic marker, synaptophysin, which is not changed in either of these, but then when you look at without METH and with METH, you see a significant increase in this particular expression of this ATP1A3. We chose 250 micromolar. We did an initial MTT assay to look at the viability of the neurons using a dosage curve for methamphetamine, and we found that at 250, and there was like about 90% viability of the cells as such. And 250 is also in the range of the meth abusers who, chronic meth abusers who at least abuse methamphetamine like 3 times a day as such. So the range is anywhere between 50 to about 600 micromolar in the blood plasma as such. So since I said with the monkeys we had only 2 groups, we further went ahead and we wanted to understand the true synergy, meaning that how does the virus and the drug sort of interact and then cause an impact in the expression of this particular protein. And here so we basically chose to use pro-inflammatory cytokines, and as you see that we basically used METH only on the neurons, interferon gamma, and then both interferon gamma and methamphetamine as such. And the other pro-inflammatory cytokine we chose was TNF alpha as such. So compared to these two, when you use a combination of, you see an upregulation of this ATP1A3, much significant in a combination of this pro-inflammatory cytokines versus METH only as such. So this further validated that indeed there is, when you have an interaction of the virus and the drug, does cause an increase in the expression of ATP1A3 as such. So we, like I mentioned in this couple of slides earlier, that there is an ever evolving role of this ATP1A3 as a signaling molecule. We wanted to understand in HAND how does ATP1A3 actually mediate its effect. So we just took rat striatal neurons. We performed a time course to look at the expression of the MAP kinase family, especially the ERKs and the JNKs. We see a nice characteristic phosphorylation pattern with methamphetamine as such, which is depicted here in the quantitation. And then this led us to thinking that MAP kinases are a big family, is there ATP1A3 very specific to any of this kinases as such? So for that, we did inhibitor studies, and we found that when you use the ERK inhibitor U1026, you basically attenuate the expression of ATP1A3 with methamphetamine, but it is not the case with the other inhibitors for J and K and for the phospho-P38 as such. This was pretty interesting as to us. And then, since this was again only in the presence of methamphetamine, we further went ahead and did also in the presence of pro-inflammatory cytokines, and we saw that this was very specific to we see an abrogation of the expression of ATP1A3 with the ERK inhibitor, and it was actually not the case with the other two. I don't have that here. So just to summarize the the organelle proteomics in HIV METH synergy, we found one potential lead, the ATP1A3, which is actually increased by both methamphetamine as well as pro-inflammatory cytokines, and the specific signaling of ATP1A3 is actually mediated by METH is actually ERK specific. And currently I am moving this analysis more into an in vivo approach using the HIV transgenic rat model, because the rats are commercially available, much cheaper than the monkeys as such, and we could also definitely look at the synergy of the virus and the drug as such. So one of the avenues that I'm really interested in looking at is the signaling aspect, because ATP1A3, it is actually present in the membrane microdomains, which are a haven for signaling molecules. So one direction that I'm currently working at is using mass spectrometry, basically identifying the potential binding partners of ATP1A3 by co-immunoprecipitation as such. So the current work that I'm doing now, so I've sort of started to get away a little bit from HIV and focusing more on drugs of abuse per se, specifically meth abuse, and more from the standpoint of the behavior, because chronic meth abuse has a significant impact, and one of the areas that I'm actually venturing is on the role of chronic meth abuse, psychosis, and how this actually alters the behavior. So what are the ongoing changes that are actually occurring at the level of the synapse? So that's the main focus. So to understand that, I'm starting to use a systems approach, where systems biology, which is a study of systems of biological components, which may be molecules, cells, organisms, or the entire species as such. So basically, in a snapshot, we want to utilize a systematic measurement using technologies such as genomics, proteomics, metabolomics, and of course in conjunction with mathematical and computational models to kind of understand the biology associated in this direction as such. So here is a little schema that I actually shown earlier. People were looking at the cell, one gene at a time, but then with the advent of high throughputs, basically we can look at the functions of the cell either at the at different levels, especially gene, RNA, protein, or metabolite level. But nowadays, with the systems approach, you can actually combine all this to get a more exhaustive analysis of the real changes that are actually occurring during in whatever the interest that you're looking at here, in this case drug abuse associated changes in behavior as such. So there are currently I am doing this in collaboration with Dr. Rick Bevins, who is the chair of the psychology department at UNL, and Kiran or Dandi Bastola, who is a computational biologist basically at UNO. This is one of the projects which just got funded as part of the state of Nebraska Research Initiative, and here we are trying to understand the impact of this anti-inflammatory drug called ibudilast or AV411 in elucidating synaptic changes in chronic meth abuse as such. The other project which is ongoing is looking at sex differences, because we just got some very nice data. I mean, in the interest of time I wouldn't be talking much, but I just want to give you a little flavor of that. We found that the female rats are actually very difficult to, the relapse that we see in female rats basically after you extinct them from the drug is much, much higher compared to the male counterparts as such. So we are trying to understand what, and the behavior is also severely different between the two sexes. So we are trying to understand from a standpoint of a systems approach, like, what are the ongoing changes at the synapse as such? How is the synaptic plasticity sort of altered, in this scenario as such? So anyway, just to touch back of this project one, AV411, as I mentioned, is an anti-inflammatory drug that modulates the activity of glial cells, suppressing the production of pro-inflammatory cytokines. There is immense potential of this drug in reducing neuroinflammation. There have been a bunch of studies where they have shown possible, potential applications of this drug in relapse-remitting multiple sclerosis. Sanjay Magirwar's group has actually shown that, the treatment of of neuronal cultures with AV411 has been shown to reduce STAT-induced neurotoxicity. And there is also the biggest boost that we actually got was this paper from Beardsley et al., where they show that this particular drug has been shown to prevent relapse in chronic METH administration as such. So the question that we actually want to ask is, how does AV411, given its anti-inflammatory impact, the synapse in METH abuse? So here there are two directions in which we are going. What are the synaptic fingerprints differentially expressed? And then what are the genes or the proteins targeting specific functional pathways which are actually involved here as such? So we are using a three-prong approach comprising of a behavioral component, a biochemical component, and a computational component, majorly with the sole focus on the synapse as such here. So our behavioral component comprises of self-administration. So this is a chamber where you have one rat basically getting a shot. It's like, very similar to the human abusers who take intravenous injections as such, So our study comprises of 4 groups. The control is the saline only throughout the entire study, and then we have 3 groups of methamphetamine which receives, and then of this, one group actually receives saline during the extinction phase. I'll be showing you it in the next slide, and the other 2 groups receive the 2 doses of this particular drug at 2.5 and 7.5 milligram per kilogram as such. So this is a this is a graph showing the amount of lever presses as such. So the whole regimen, we start off with an FR1, a fixed ratio 1, where the animal has to actually press a lever to get a shot of methamphetamine as such. So we then make it a little difficult, because drug abusers, they don't get the drug on and off, they don't get it all the time. So we kind of make the task a little tough as well, increasing it to 3 lever presses over the course of 5 days. And then from there onwards, we maintain it at FR5 as such. And again, mind you, this is something very new to me. I'm just learning the basics of the behavioral biology as such. And then the whole regimen lasts about a little over a month, where after that, we start giving them, the extinction phase, meaning that the animal, despite pressing the, like I classified them, the salines, and then one group of methamphetamine is getting only this particular, just the saline, and the other 2 gets the drug as such. So in the interest of time, here is the little graph that we shows for the 21 meth treated animals, showing the different amount of meth they have actually taken. And then here, this is the graph showing that, we basically could bring down the craving that was associated with meth using, this particular drug, especially 2.5 and 7.5. They actually show a very nice sensitization here as such. And currently, what we are doing is we have isolated the prefrontal cortices. The reason we chose is because PFC is associated neuroanatomically with the striatal and the limbic structures, and more importantly, our major focus is on understanding behavior. PFC is also associated in decision making, regulation of emotions, and in drug addiction. And more important, it can be obtained in larger amounts to yield ample protein as well as RNA samples as such. And then we are currently doing microarrays as well as quantitative proteomics on these. And of course, we will be requiring the support of computational bioinformaticians as such. I think I'll probably skip through these slides to understand. And then this is generally how it looks like when a computational biologist gives us about the interacting pathways as such. So it's like a nice brightly lit sky with lots of stars in it, but this is the interactive partners that we actually need to understand what are current involved or what are differentially expressed as such in these. So we use a approach called as correlation network analysis, where we try to correlate the gene expression data to the protein expression data. So basically to build functional pathways and basically identify those genes of proteins which are potential targets, and then we go back to the bench to validate them through an array of biochemical and other molecular biology approaches as such. So here I just wanted to touch base that, given the fact that microRNAs are also very important regulators of synaptic plasticity, this is a very, very preliminary data from human brain tissues where we isolated microRNAs from the synaptosomal fractions, and then we did a analysis and we found one of the microRNAs MIR219, which is very significantly enriched in the synaptic fraction as such. So again, these are all from from individuals who passed away and had no case history. These are again control subjects. So we are yet to look at the expression of these particular things in a diseased context as such, and that's one of the other ongoing. The reason I highlighted 219 is that it basically has been shown to modulate NMDA receptor mediated neurobehavioral dysfunction. It's been implicated in schizophrenia, and it looks like we have something which we could potentially look at METH associated CNS dysfunction and its potential implications on behavior as such. So just to tie it down, oops. I don't know, it's not going back. Okay, just to tie it down, the whole point what I'm trying to say is that we are applying, systems biology can sort of be summarized in a nutshell, where you basically have an integrative analysis at different levels, gene, RNA, protein, use integrative analysis, computational analysis as such, build these functional clusters, which then as biologists to us will be further valuable in understanding their role, specifically from understanding mechanisms as such, both in vitro and in vivo. Anyways, I'll just probably skip this one, and just to summarize my talk, I've tried to show you that there is very huge potential of post-genomic strategies which have been profound with advanced genomics-based strategies. The real changes are actually in the brain. Major emphasis has been on the synapse, and integrating omics strategies will better lead to understanding the physiological alterations, and in this aspect, a systems biology is one of the very promising avenue to gain potential insights into these neurological conditions. So with this, I would like to acknowledge the people with whom I've been working. I was having I would like to thank my postdoc mentor Howard Fox, my collaborator Soumya, who is an expert in microRNAs as such, James Busher, my technician, who's helped me with the synaptosomal preps, my current collaborators, Rick Bevins, Lloyda, with I really look forward to with our work on acute HIV infection, my mass spec collaborators Gary Siusdak at Scripps, Sunia Traver at Harvard. We are currently supported by two wonderful bioinformaticians, Babu Guda and Kiran, and then of course the funding from the state of Nebraska in this rough climate of funding is really appreciated, and thank you so very much.